In this laboratory, we study cell skeleton. Indeed, cells have a skeleton. It is dynamic, permanently self-assembling and disassembling in a global turnover which allows cells to control and change their shape. We developed a new technology to constrain cells in regular geometries, squares, triangles, in order to manipulate and study their intracellular architecture. Recently, we have even been able to open cells and purify the filaments of their skeleton in order to let them grow alone on these patterns. For the NUIT BLANCHE, we decided to move the lab in the street and turn our squares and triangles into a more complex structure, a building. We wondered how a single cell would behave in a maze with the shape of a building in which windows would serve as obstacles, and if a group of cells would solve it differently. We also wondered how they would react if, on the opposite, they would be constrained to attach to windows. We could then look in the cells and see how their architectures adapt to the one of the building. We found even more challenging to perform experiments without cells, with only isolated filaments, which would be forced to grow out of the windows and doors of the facade of the building. We wanted to see the structures they would form spontaneously in the building, as if the latter was a gigantic cell. First, we miniaturized the building. To that end, we digitalized the shape of the building. We then used a photosensitive resist on which we projected the picture of the building through the objective of our microscope in order to polymerize a master. This master was further used as a mold to cast an elastomer, which was baked, and the working area was cut out to obtain a maze with hundreds of mini buildings. In the lab, we work with numerous distinct cell types. We chose cells from the retina, from bones or blood, which were first put in culture in flask, then aspirated and plated on the mini buildings. For the other type of experiments, we made complex chemical mixtures based on numerous isolated cell components. The mix was then injected on the mini buildings to let the filaments grow from the facades. We then used specific microscopes for each experiment in order to record cells and filaments. These structures grow and move slowly. They were recorded during hours and days and then accelerated to make movies. Finally, those movies were projected onto the original building in the center of Paris.